Hey, welcome back. Hope everybody enjoyed their weekend. Somehow last week felt super long, even though it was only a couple days. Hopefully you feel refreshed. Okay, so we're gonna finish up 1 second with the concept of homeostasis before we break into our review of biomolecules. Okay, so when we left off last time, and we were thinking about homeostasis, we came to understand that in physiology, in your bodies, we have a set of regulated variables that makes your body tries to keep consistent, right? It's important to the function of your body. It's important to keeping you alive. Right. So these regulated variables have set points. So there are specific values we want to keep them around. And when they start to vary from that set point, so when they start to go off, whether it's up or down, we have sensors that tell us that information. We start to produce what we call an error signal. So to maintain homeostasis, through the process of negative feedback, we pull those regulated variables back towards their set points, right? So we're reducing that error signal, just moving in the opposite direction back to where our kind of like normal baseline is. So we thought about the concept of negative feedback. Since we thought about the concept of negative feedback, it's important to note that positive feedback does exist. And it's kind of great that this is the thing that we didn't get to though, because positive feedback is usually not too involved in the process of maintaining homeostasis as much as it is involved in like very specific biological processes that happen for very specific scenarios. I have one example on here that, roundabout kind of way does sort of maintain blood volume. So one place you would see positive feedback is when we see blood clots forming. I think thinking about positive feedback versus negative feedback is helpful to do in kind of picture form, right? So for our negative feedback, we're thinking about over time, our regulated variable may change in one direction, right? And then negative feedback pulls it back in the opposite direction, so a negative reaction, whether that opposite happens to be pulling it back down, or whether that opposite happens to be pulling it back up, right? The opposite direction of the arrows is the key thing to focus on for negative feedback. If we're thinking about positive feedback, we'd still be looking at something happening over time, but you want to think like a snowball effect for positive feedback. So if you think about blood clotting, right? So think about like amount of clots or something here, right? So positive feedback is going to be when we see a change in one direction, spurring more change in that same direction, right? So instead of pulling yourself back down to a baseline value, you're doing more and more and more of that same thing. So hopefully, hints to why this isn't so useful for homeostasis, right? Homeostasis is about maintaining something constant, right? So when we have positive feedback, we're actually making a change more and more and more rapidly. So positive feedback is sometimes important in addition to thinking about clotting. Another place that we'll see positive feedback is when we think about the reproductive system, we'll see some positive feedback there. Maybe we'll see it a little bit with the function of nerves. We'll talk about that when we get there. It's not generally seen in homeostasis, but it's an important thing to know it exists, so you know what that word means when we refer to it in specific units. So here's an example of what a positive feedback loop would look like. So usually these dotted lines in diagrams and in textbooks are going to indicate negative feedback. So usually they're going to be telling you that we're turning off a signal, right? And the arrow split. But in terms of positive feedback, what this is saying is that your pituitary gland, hanging down from your brain, produces a hormone called LH, which makes your ovaries secrete estrogen. And we have a positive feedback loop here. What that would mean is they produce more and more and more estrogen once we first see that estrogen in the system. So this is a rarer form of feedback loop, but we'll see it sometimes. The final sort of thing to be aware of as we think about general big picture types of processes involved with maintaining homeostasis is that when we think about regulation of the body throughout this course, we're often going to separate thinking about things that are intrinsically regulated versus things that are extrinsically regulated. So when we say something intrinsically related or intrinsically regulated, we're not just saying that it's regulated in your body. Both of these processes would be in your body. What we're saying is that it's happening really locally. So if we say something's intrinsically regulated, we're saying like we're regulating the heart by seeing changes directly in the heart cells communicating to each other, right? So super local signal. When we say something's extrinsically regulated, this is when we think about the brain usually, but generic term kind of integrating sensors. This is when we're going to have a signal from some other organ system involved in adjusting and regulating that process. So if your heart is being regulated by nerves coming from your brain, right, that would be extrinsic regulation of your heartbeat. And we'll usually see within each of our organ systems that these are working together, working in conjunction. So we'll see some effects happening locally, so some intrinsic regulation, and some effects happening in response to systems outside that organ, so being regulated extrinsically. So usually that's going to be involving nerves, the nervous system. Sometimes it'll be using the endocrine system, so hormone space. So we're going to move on to our next set of lecture slides. So what we're going to do here is we're going to be reviewing our different types of biomolecules. Probably Wednesday we'll get to enzymes, but maybe a little faster. Okay. So what we're going to be thinking about is what makes a biomolecule in the first place. We want to make sure that we remember the different classes of biomolecules. So we're going to be focusing on 4 of them. And we want to make sure that we understand how to tell which one we're looking at, but more importantly, how they function and how they function differently. Because that's going to be important for our cellular processes. That's going to be important for understanding membranes and their behavior. It's going to be important for understanding what can and can't get transported across cells under what conditions. So this is kind of the baseline for what we're going to be doing throughout this course. And we're going to talk at the end about a specific type of protein, the enzyme, and how that works in chemical reactions. So when we're thinking about biomolecules, you may also have referred to them as macromolecules because they're generally kind of big. We're thinking about molecules that are biologically synthesized, so made by living things. So they have some things in common. So one particularly noticeable thing in our biomolecules is going to be that they have carbon in them, right? So carbon has a basis for life. We've got carbon in our biomolecules, our organic molecules. Other atoms that we'll see commonly include oxygen, hydrogen, and nitrogen. Those are the main ones to focus on, although we'll see other trace elements. Phosphorus will play a big role in a couple of these. Yeah. So mostly carbon, oxygen, hydrogen, nitrogen. So we're going to break this down into 4 basic types of biomolecules, each of which will have some subsets. So we're going to first talk about carbohydrates, and we're going to talk about lipids, and we'll talk about proteins and nucleic acids before zooming in specifically and thinking about enzymes. Starting with our carbohydrates. So when we're we're thinking about a carbohydrate, we're thinking about molecules that look like these on the board. Right? So they're composed of carbon, hydrogen, and oxygen atoms. And they can take different shapes, but they're going to be vaguely ring-like in our bodies because we're in a liquid environment. So they're going to come in a number of different sizes as well. So a monosaccharide is going to be our monomer of our carbohydrates. We'll think most often about glucose. So here you can see glucose, right? We're usually going to have a 1 to 2 to 1 ratio, pretty much always. That's kind of our rule for carbohydrates, right? Whether we're a monosaccharide with 1 subunit, a disaccharide, so sucrose is table sugar, so 2 subunits. We can mix and match our different monosaccharides to build disaccharides, or big polysaccharides, so poly meaning many, right? Here we have an example of glycogen, which is an important storage molecule, storing glucose in your body for later use so that you can have glucose to break down for energy even when you're not eating. So these are some glucose units within our big glycogen molecule. If we're thinking about like food that comes from plants, starch would be the corresponding large polysaccharide that you find in plants. So if you eat something really starchy, what you're saying is you're eating a bunch of this big storage carbohydrate. If you're looking at a detailed diagram where we have all the atoms listed out, you're making sure that you're looking for carbons. So we're going to remember that we have carbon when it's listed, but also when we have a point where things come together. If there's no atom listed, we're just assuming that's a carbon because carbon really forms the backbone of all these molecules. It's a safe assumption. So we're looking for our carbons, we're looking for hydrogens, and we're looking for a roughly equivalent number of oxygens to those carbons, right? Should be the same, 1 to 2 to 1, equal carbon and oxygen, double the hydrogen. So when we think about carbohydrates, we can think about how we make them, and we can think about how we break them down. In particular, we're going to be thinking about a very specific form of breaking down carbohydrates when we think about metabolism and we start to think about how we make ATP, our cellular form of energy. But for right now, we want to notice about our carbohydrates that they're linked together with covalent bonds, right? So pretty stable bonds here. When we form larger carbohydrates from our monomers, our monosaccharides, here we see a glucose and a fructose. We see the formation of sucrose. You can see that this is a condensation reaction. So what that means is that we are forming water as we form a covalent bond, so a stable link between these 2 monosaccharides. So you can see the relevant groups here that were coming together, right? And we can see that in our final configuration, we're keeping 1 oxygen here in the center, and we can see there's now a covalent bond to our glucose, there's a covalent bond to our sucrose, which means that we've lost a couple things, right? So we've lost on here 1 hydrogen from one of our monomers, an oxygen and another hydrogen, and that's why we just have that 1 oxygen left. So those have come off in the form of water. So that's our condensation reaction. I'm pointing this out really because I think that carbohydrates are sometimes particularly recognizable because they kind of look like they're holding hands. If you think of that O in the middle, it's kind of like a fist, like 2 hands grasping each other. And that's happening because of this specific way that they form. Because we're going to see some other types of macromolecules that also have rings, but you're not going to see other types of macromolecules that are linked together quite like this. This also means that when we split up carbohydrates, we're going to be adding water, right? So we got to get those 2 hydrogens and that oxygen back in order to break back into monosaccharides. So this is going to be a hydrolysis reaction. So hydro for water, lysis for breaking stuff up. So larger saccharides, so disaccharides, polysaccharides, get put together when you add water. And you can think about making like sweet tea or something. So you've done this probably yourself in your kitchen in some form, right? You take a mug of tea or a glass of water, add sugar to it, stir it up. You see that sugar dissolve, right? Can take a while depending on the temperature of the water that you're using, depending on the system, but you can see that water breaking apart our carbohydrates. Example. Okay. So this is because carbohydrates, one of the things that we're going to notice later on, so I'm going to point it out to you right now, because of all these hydroxyl groups, right, all these OH groups, they're going to be hydrophilic. They're going to play well with water. They're going to mix well with water. They're going to dissolve with water. All right. So before we get into our next topic, I'm gonna quickly check what you remember about biomolecules, macromolecules as relates to cells, because we're going to start to get into that a little bit as we talk about this topic. So what do we remember being a particularly important molecule, kind of our baseline of cell membrane? Okay. So when we think about cell membranes, we are going to think about a number of different biomolecules being involved, but our phospholipids are going to be basically our base layer of the cell membrane. Our phospholipid bilayer forms the main part of the structure of a cell membrane. And so that's basically what's making the bubble. Proteins are also important, particularly because they allow transport. Carbohydrates are there. Usually they're involved in a little bit of signaling and cell recognition, but the phospholipids are going to be our kind of fundamental basis of the membrane itself. Backing you that, because what we're going to talk about next are our lipids. So this is our second class of biomolecules that we're interested in. Okay. So lipid is going to be mostly made of carbon and hydrogen. Carbohydrate had carbon, hydrogen, and oxygen in a 1:1 ratio. Lipids, we're not going to focus on a specific ratio. It's going to be carbon and a lot of hydrogen. Now this is important because that lack of oxygen kind of changes their behavior, right? So oxygen in general tends to cause some polarity. So basically, when we think about, you remember concepts of electronegativity, right? We have different atoms that attract electrons in different amounts, and oxygen tends to like really pull them. Okay? But carbon and hydrogen, they're kind of an equal pull, right? So they share much more equally than they do with oxygen. Okay? So because our lipids are just hydrogen and carbon, not oxygen, sharing those electrons equally means we don't have any polar behavior here in our lipids. And that's important because, like our carbohydrates, even more than our carbohydrate, water is a polar molecule. Okay? So the fact that water is polar but lipids aren't means that lipids aren't going to mix very well with water. So lipids are non-polar, so they don't have like little charges accumulating on them because of little differences in how long the electrons are spending around the specific atom. We're if you remember the word dipole moments, right? They don't really have those. If you don't remember that word, I'm not going to ask you about it later, but if you do, that's what we're talking about. Okay. So they're nonpolar, they're hydrophobic, they kind of run away from water. A classic example of a lipid is like an oil or fat. So picture mixing together oil for your lipid into some water. They're just not going to mix together, right? They're going to separate out. You're going to see little bubbles of that oil, little bubbles of that fat separating out from the water. That's because of this non-polar quality that leads to lipids being hydrophobic. And that's really what unites our lipids together, right? So that's going to be true for all of our lipids, really. Okay. This is important to note because there are 5 kind of subclasses of lipids, but they would all be made of hydrogen and carbon, and they'd all, in general, be nonpolar and hydrophobic. Okay, so for those 5 classes, we got triglycerides, ketones, phospholipids, eicosanoids, and steroids. The ones that you should consider most important, triglycerides, because they're like the classic example, are going to be important to look at the structure of. Their structure is also going to help us understand phospholipids, which are like our #1 most important kind. All right, so I'm going to say 1 for phospholipids, 2 for triglycerides. If you want to add on a third one, a bunch of our hormones, our steroids, cholesterol, which is important for your health, is a steroid. So we're going to make steroids #3 in terms of importance. So be aware of these words, be aware that there are types of lipids. So in terms of what you want to concentrate on, recognizing and making sure you know, going to be those 3. So we're going to look at a triglyceride first, because that's our classic example of a lipid. So it's called a triglyceride because it has 3, so tri, fatty acids. The fatty acids are these big yellow chunks that we can see on the right-hand side, okay, attached to a glycerol backbone. So that's the glyceride part. So if we just had the glyceride backbone, this wouldn't be a lipid anymore. This would be a 3-carbon alcohol, right? But because we have these 3 fatty acids, that's what makes our triglyceride a lipid. You can see that a fatty acid chain is really just a ton of carbon and hydrogen linked together. A long hydrocarbon chain. Okay. So that's our general form of a triglyceride. I often, when I'm drawing them for myself, in case I do this later, tend to draw them just kind of like a flag, right? So glycerol backbone with our fatty acids coming out to the side. So if I do that, that's what I'm drawing. Now, we do have different types of triglycerides, which you probably noticed if you look at like a food label, because food labels will separate out sometimes our saturated fats from our unsaturated fats, even with our trans fats as well, because different types of triglycerides are processed differently by your body, so they have different impacts on health, basically. And the reason for that is their shape. Okay. So when we're looking at a saturated fatty acid, what we're saying, what it's saturated with, what it's full of, is hydrogen. Okay? So when I draw a squiggle, and what they're abbreviating here in that hydrocarbon chain, so we've got our carbon backbone, right? Got our carbons, right? And we've got hydrogens coming off of them. So I'm going to ignore where we're linked to our glycerol here. Okay. But a saturated fatty acid is going to have a hydrogen everywhere you can put one. So carbon is going to make 4 bonds, which means right here we have space for 2 more, so 2 hydrogen. Same thing here, our second carbon. Same thing here on the third. And then at the end, we've actually got room for 3. So that's what we mean by saturated. So saturated fatty acid is full of single bonds saturated with hydrogen. Okay. Now, an unsaturated fatty acid. Unsaturated meaning it could hold more hydrogen and it's not. It's going to be unsaturated because we're going to see a double bond somewhere in the chain between two of the carbons. So if we have a double bond between two carbons, right, carbon can only have 4 bonds, but you'll notice the problem. Right now I have 1, 2, 3, 4, 5 around each of these two carbons. So the result is that we lose a hydrogen from each of those carbons. So that's why they're unsaturated, right? We had to take away some of that hydrogen. So that's an unsaturated fatty acid. And adding one more layer of detail to that, our unsaturated fatty acids come in either trans or cis form. And what that's describing is the position of our remaining hydrogens, right? So you see on this side, I've left 2 hydrogens on the same side of those carbons, on the same side of that chain. So this would be a cis fatty acid, okay? So same cis. A trans fatty acid still has a double bond, but the position of the remaining hydrogens are trans across on opposite sides. So those are our different types of fatty acids. You might see any of them in a triglyceride. So that's why we have trans fats. What that's telling you is that in those fats, we have a double bond in the hydrocarbon fatty acid chain, and the hydrogens happen to be positioned on opposite sides. That's what we're describing here. That's what a triglyceride looks like. Our next lipid, so that we can be completionists and actually name all the different types of lipids, are going to be our ketones. So ketones will come up and be important in diseases like diabetes, or you're on the keto diet, on a particularly low carb diet, you may be producing extra ketones as well. And that's because the main way you end up with ketones in your body is through the hydrolysis of triglycerides in adipose tissue, aka the breakdown of fat, right? So if you're breaking down fat in a hydrolysis reaction, adding water, right? So breaking down fat instead of breaking down carbohydrates, you're going to release fatty acids. So we have those 3 fatty acid chains on the triglyceride. And some of those fatty acids are going to go into the liver, and the liver is going to modify them into this structure called a ketone. So you can see an image of what a ketone looks like here. So I'm kind of specialized. I probably wouldn't show you a picture of a ketone and expect you to know that it's a lipid, but you should know that the word ketone is describing a lipid. So when we're thinking about ketones, we think about potentially some similar words like ketosis or ketoacidosis as a buildup of these ketones. And it's something you can actually kind of smell. So you smell someone who's undergoing the ketoacidosis, whether they have diabetes or whether it's diet, you could potentially smell that kind of coming out in their breath, something that smells a little like acetone. Okay. Our number one most important type of lipid is the phospholipid. So let's spend a little time with that. Okay. So when we're thinking about a phospholipid, we're going to have part of our phospholipid that has this typical non-polar hydrophobic characteristic that we expect from lipids. We're going to have another additional part that has a little different behavior. It's a polar kind of behavior that's going to be hydrophilic. It's going to be attracted to water. Okay. So typically, when we're thinking about our phospholipids, you're going to have your polar phospholipid head, right? So P for phosphate and P for polar, that's kind of handy, right? So polar phospholipid head. And then we're going to have our non-polar tails. There'll be 2 of them. And they're going to look just like the tails on the triglyceride, except there's 1 fewer, right? So here's our polar part of the molecule. So attracted to water, polar and hydrophilic. And here's our nonpolar part, just composed of carbons and hydrogens. So this is our nonpolar part. It's going to be hydrophobic, it's going to repel water and anything that likes water. So we refer to phospholipids as being empathic because they have these two parts. That's what they mean. It has two parts with these different behaviors, right? These different attractions or repulsions with regards to water. I'll put some on here. Right. So here's another drawing of a phospholipid. So we have that hydrophilic head attracted to water, it's polar. We've got those hydrophobic tails, they're repelling water. They're just typical hydrocarbons. So just like the fatty acid tails on a fat burning oil. Now, this amphipathic property, this property of having one end that's attracted to water and one end that's not, means that if you drop phospholipids in a glass of water, or if you drop them in a watery environment like your body, right? We talked about total body water and how basically all the cells of your body are surrounded by water. And phospholipids in water automatically orient to create specific structures, right? So we're automatically going to see those fatty acid tails that are nonpolar and hydrophobic run away from water. And we're automatically going to see the phosphate head, polar and hydrophilic, orient towards water. Okay. So I'm going to mention the less common example first. So one way we could do this is have all those fatty acids cluster all together on the inside with all the phosphate heads on the outside. So here are the tails, here are the heads, polar and nonpolar. Right. This is what's called a micelle. We'll briefly mention them again in A&P2. They've got a while before those come back. But we're going to see the other form all the time, like every day in the class. So the other form that phospholipids can orient into is a phospholipid bilayer. Okay. So if we remember, when we're talking about our total body water, right, two types of our compartments or our types of fluid were our extracellular fluid and our intracellular fluid. Okay. Right. And because we have this water outside the cell, extracellular fluid, and water inside the cell, intracellular fluid, this basically means we have two watery regions that we need to protect those fatty acid tails from. So that's why we have a phospholipid bilayer, right? So bi is two, right? So what's happening here is we've got phosphate heads lining up along the extracellular fluid. We've got phosphate heads lining up against that intracellular fluid. And then the fatty acid tails of each extending towards the center of our membrane, right? So the end result is that we're going to have what I kind of think of as like three sections to a membrane that's made up of phospholipids, right? So we've got a polar part. It's relatively thin. Then we have a really big nonpolar region because of those fatty acid tails and the fact that they're doubled. So let's even say multiply by 2, right? So 2 times nonpolar. Don't worry about the numbers if they don't make sense, but just notice that this is a really big layer. And then we've got that polar inside again. So when we start to think about membranes, we're really going to be focusing on that big, thick, nonpolar, hydrophobic region that forms in the center of the cell membrane. And this can continue all the way around. It would just take me a long time to draw, but all of these would be lined up kind of foot to foot. So a typical example of a phospholipid bilayer is the cell membrane. But if you're curious about where we might see in my cell, one of the places you'll see those is kind of in your GI tract as you are kind of trying to digest different types of triglycerides and stuff. Occasionally we'll have these starting to form from collections of whatever you ate. Another place we see a phospholipid would be in your lungs. So when we talk about the respiratory system, also in A&P too, we'll note that this phospholipid in your lungs stops them from collapsing in on themselves. So our phospholipids are important for the function of every cell, and we're also going to see specialized kinds doing specific things within specific organ systems. Our next type of lipid, again, be characterized by being mostly made of carbon and hydrogen, big hydrocarbon chains, nonpolar, hydrophobic, are the eicosanoids. So these are a modified kind of fatty acid, and when I say modified, basically you notice that instead of being just one long chain, kind of like turned on itself, right? So we're talking about this link here. We're not going to see these often in this class, but they do exist. Usually what they're doing is functioning in intracellular communication, so kind of working in conjunction with hormones, basically. When we think about hormones, we're going to be thinking about like lipid types and protein types. So examples of some eicosanoids that we'll see later in this course potentially would be prostaglandins and thromboxanes. But mostly we're going to be focusing on the other types of lipids. But I know some people are really jazzed about chemistry, so worth taking a peek. Next, we have our steroids. Okay, so steroids are another place we'll see kind of a ring-looking structure, but we're going to make sure that we know this isn't a carbohydrate because we don't have all that oxygen. Right? We don't have oxygen bonding together those rings, and we really don't see much oxygen in the structure at all. We might see oxygen in chains coming off the side somewhere, but it's not forming like the fundamental block of the steroid. Okay. So all steroids are going to be derived from cholesterol, which is why they all have this kind of four-ring structure with these rings kind of pressed against each other. So some common steroids that we'll see are steroid hormones like estrogen and testosterone, sex hormones, aldosterone that we'll see later on as well when we start to think about kidneys and different types of regulatory functions in the body. That brings us to the proteins. So proteins are going to be really large biomolecules. They're kind of the most macro of macromolecules. So they're going to be big, long polymers. So polymer, poly, many subunits linked together. So big, long chains that get folded together. They're going to be made up of amino acids. So our monomer of a protein is an amino acid, so that's our building block. Okay. So there are 20 different kinds of amino acids. So we're not going to be too specific about which ones are in the protein examples that we look at right now. But what's important to realize about that, there are 20 different choices for amino acids. It means we can make a lot of proteins that are really different, right? We have so many different choices for amino acids, which means we can have really different chemical chains when we're building a protein, which means we can have really different shapes when we fold up those proteins. This is important because proteins basically do everything in your cell. They provide some like kind of physical scaffolding, so some structure. They're going to transport molecules in and out of the cell. They work as signals between different cells. They're really important to how your body functions, and they can do so many things because we've got so many different building blocks. Okay. If we're building a protein from an amino acid, we're going to start by linking them together. So let's peek at these amino acids for a quick review. All right, so for our amino acids, you're going to notice that regardless of which one we're thinking about, it's going to have a central carbon. So we've got this carbon in the middle, alpha carbon. We're going to have an amino group on one side. So here we see our amino group. So amino, think about that N in amino, because an amino group has a nitrogen. Right, so we've got this nitrogen containing amino end. And then on the other side, we have our carboxyl group, right? So we're thinking carb for our carbon and oxyl for our oxygen and our oxygen and hydrogen down here, right? So our carboxyl group is this one, carbon double bonded to oxygen on one side, and a hydroxyl group, so oxygen and hydrogen on the other side. At the bottom, we can see there's just a little hydrogen hanging out. And we see this R group here up top. Okay, so this R group, notice it has a 1 next to it. So the R is not standing for any specific element. What the R group is I like to think of R as like replaceable. So the R group is what's going to be different between each of those 20 amino acids. So all 20 amino acids are going to have an amino group, a carboxyl group, a little hydrogen, and that central carbon. So the R group, you got 20 choices. That's the piece that makes all the amino acids different from each other. So when we start to link together our amino acids, right, we're going to take the carboxyl group from one end and the amino group from the other end, and we're going to link them together. And that process is going to release water. So this is also a condensation reaction. So you can see from 2 amino acids, when we link them together, here's where we formed a bond between them. Right? So here we've got a covalent bond between that carbon from the carboxyl group and the nitrogen from the amino group. So that's referred to as a peptide bond. The peptide bond is the covalent bond that you're going to see linking together amino acids. Yeah. When we're talking about amino acids chained together in a line, before we have any folding, we're going to refer to them as a polypeptide. So that peptide word, every time you see the word peptide, think protein. Got something to do with proteins. So here we have a little peptide. So if we're calling it a peptide, that usually means we've linked together like 2 to 50 amino acids. We can link together more as we start to form larger proteins. So our big proteins are going to be more than 50 amino acids for sure. Let's get our first level of structure. So proteins have levels of structure. So we're thinking kind of like steps to building a protein. So the primary level of structure, the first step, is linking together those amino acids with our peptide bonds. Right? So here's our primary structure with those peptide bonds forming, right, giving us our polypeptides. Right? So we're going to notice in the chain of a polypeptide, as we're getting towards building our protein, you're going to see a nitrogen, then 2 carbons, and a nitrogen, then 2 carbons, and a nitrogen, and 2 carbons over and over again. Right? We didn't see nitrogen in our carbohydrates. We didn't really see nitrogen in our lipids. So if you see a ton of nitrogen, got a hint, maybe we have a protein that we're looking at here. Okay. Our second level of structure is as we start to fold this chain. Right? So we've got our polypeptide chain with those covalent peptide bonds all in line. And then we start to twist in different ways, or maybe fold a little bit. Okay, so our secondary level of structure, the reason this is happening is that the hydrogens and oxygens left from the amino groups and those carboxyl groups, if they come kind of near each other, they form hydrogen bonds. which is a pretty weak-ish, at least compared to a covalent bond, a weak kind of association, but you're thinking like a little bit of a kind of magnetic action. Right, so when they come near each other, we get these hydrogen bonds, and we form two specific shapes, either alpha helices. So alpha is really just A in Greek, right? So an alpha helix, A, that's your first option. Making a little curl. Or our beta, which is really just a B, right? Our beta pleated sheet. So pleated sheet, we're thinking like a pleated skirt. This is a kind of accordion fold situation. But you can see that this is also forming from the same type of force. We've got our hydrogen bonds here, right? So weak-ish compared to covalent bonds, attraction between our amino acids that don't happen to be right next to each other. Tertiary structure of a protein is as we start to crumple this together. So when a polypeptide, so this peptide bond, starts to form a structure, it doesn't just make one nice alpha helix, one like perfect ringlet down the whole chain. We're going to have little chunks that are alpha helices. We're going to have little chunks that are beta pleated sheets. Maybe we've got some regions that didn't form either of those types of structures. But our R groups that were hanging off those amino acids do interact with each other as well. So we saw hydrogen bonds forming those two really predictable shapes, the alpha helix and the beta pleated sheets. The R groups, we have so many choices, and they're in so many different regions of our chain that I can't predict them. A computer model probably could. A lot of different kind of crumpling. So I think crumpling up a ball of paper is going to be your tertiary structure. That's our third step. Our final step for building a protein, our quaternary step. So quaternary structure, think like quarter, or 4 quarters in a dollar, or quarter is a fourth, right? So these are really just talking primary, first step, secondary, second step, tertiary, third step, quaternary, fourth step to building a protein. Okay. So quaternary structure is different subunits coming together. So here we're looking at hemoglobin, which is a molecule that carries oxygen in your blood. Okay. And they're trying to show you by that like subtle shading, the difference in space. So we actually have 4 subunits. So this would be one piece that your body would make as a single chain and then fold up in that primary, secondary, tertiary structure. Here's a second subunit. And then at the back, we have 3 and 4 as well. You don't have to number them in any specific order. I'm just pointing out there are 4 of them. Okay. And those 4 different pieces are also attracted to each other based on what R groups are hanging out in these kind of like neighboring regions that can get associated. So this is a typical structure to hemoglobin, but things can go wrong with this as well. If you have sickle cell anemia, one of the things that has happened is we've substituted in a different amino acid, and because of this kind of domino effect of levels of protein structure, you end up with a kind of crystallized hemoglobin that's a sickle shape, so like kind of a crescent. So we're going to end there for today with those levels of protein structure. And next time we'll pick up with our nucleic acids.